This is the third video podcast in biochemistry and the last. It talks specifically about DNA, DNA mutation, the genetic basis of disease, and the modification of proteins and enzyme structures, and the structure and hydrolysis of ATP. The assessment criteria asks us to understand a simple model of a structure of DNA, talk about amino acid sequencing, talk about mutation, talking and discussing the genetic basis of disease, how modifications to a protein can result in a new structure or function, and to describe the structure and hydrolysis of ATP and its use as an energy source, as well as the effects of some specific metals and their effect on humans and mammals' health. You should by this time be able to identify the structure of proteins, and that should have come from our study during the last podcast of how the alpha helix was created or the beta plated sheet through hydrogen bonding, disulfide bonding, van der Waals forces, along with some ionic bonding and the polymer nature of proteins. Now, in DNA, we have two strands running in opposite directions. They have a base that link the two strands, and that base is positioned at a right angle to one another. They're linked by hydrogen bonds, as we saw in the last podcast, and the base pairs fit into a space between the two backbones so that the helix has its representative shape. Now, there are only 4 DNA bases, but there are 20 amino acids that can be used for proteins. So groups of 3 nucleotides form a codon that specifies which of the 20 amino acids go in the protein. So doing a little math, a 3-base codon yields 64 possible patterns, which is more than enough to specify 20 amino acids. So since there are 64 possible codons and only 20 amino acids, there's going to be some repetition. But it may require anywhere from 100 to 1,000 codons to describe a given protein. Each gene also has codons to designate the beginning and end of that gene. DNA mutation comes about as a change in the original DNA sequence. Oftentimes, these changes are corrected by the cell. They can be caused by random chance, but also by different chemicals or external conditions. So, because DNA is the driver or the template of messenger RNA, changes to the base structure in DNA will likely cause a change in the primary structure of the protein. The following video will give you a humorous look at gene mutation.
What do you think of when you think of the word mutation? Do you think about X-Men and some really awesome ability? Or the Ninja Turtles? We loved the Ninja Turtles when we were little. We might be showing our age a little bit. But mutations really are not that glamorous. Most people understand that a mutation is some kind of change. It is a change of genetic material, and more specifically, a nucleic acid. RNA and DNA, they're both types of nucleic acids. So, how does a change happen? Remember that in DNA, the base adenine A goes with the base thymine T, and the base cytosine C goes with the base guanine G. And that's all good, but what happens if the wrong base matches up? Many things can cause an error like that. There can be external factors like chemicals and radiation, internal things too, like something goes wrong during DNA replication, during interphase. Interphase is a stage that prepares cells to divide during mitosis. And now, while these things can increase the chance that a mutation is going to happen, it's important to understand that mutations, they're random. If a mutation is going to be a helpful thing for an organism, which is rare, very few mutations are actually helpful, it can't just will itself to get that mutation. An organism can't just will itself to get this. It's definitely not like X-Men either. And more about that when we get to natural selection. Many mutations are actually neutral in their effect, meaning they neither help nor harm an organism. And some mutations are harmful. So we're going to talk about the different kinds of mutations. First, we're going to talk about gene mutations. So DNA makes up genes, and genes code for proteins that influence or make up different traits. So when DNA has changes, otherwise known as a mutation, then different proteins can be produced, and this can affect an organism's traits. So let's look at the gene mutations. So first, substitution. That means you have the wrong base matched. So instead of A with T, you could put A with G. Scandalous. You can have insertion. That means an extra base is added in. You can have deletion. That means a base is removed. Insertions and deletions have the potential to be especially dangerous because remember, in protein synthesis, we talked about how bases are read in threes. Well, if you add a base or remove a base, suddenly the number of bases total has changed, right? So if you read in threes, depending on where it happened, everything that's read afterwards has the potential to get really messed up. We call this a frame shift. Now, these were all types of gene mutations, but
Now, in the video, you saw the examples of gene mutation. Now, a single change isn't uncommon and may not always cause a change in the function. If the mutation results in a different amino acid being put into the protein, the effect may still be small. However, if that mutation affects a start or stop codon, it's more serious. And deletion of a base is worse still. Sickle cell anemia, cystic fibrosis are both alterations to deletions of bases. You can see the different kinds of mutations at the bottom of this slide. If you want to stop and look at those four, stop the podcast, look at them a little bit more closely. Sickle cell anemia are red blood cells that have a crescent shape rather than their typical oval or circular shape. They're caused by a single mutation in DNA coding. You can see the sickle cell chain and the difference between a normal chain and when a sickle cell chain has been replaced by mutation. Cystic fibrosis, another mutation of a protein, affects the lungs, pancreas, and sweat glands. Instead of a watery fluid coming out of a cell, a thick, sticky mucus comes out. And in this case, three bases are deleted, and it causes difficulty for water movement in and out of the cell with the formation of a mucus. Now, some modifications to a protein and an enzyme can have beneficial results. And for example, biofuels are made from woody plants. Now, the woody material, lignin, you see that woody material in the slide picture down at the right, prevents easy extraction of what's inside the cell to make biofuels. Now, scientists in the USA have modified a protein to reduce the amount of lignin in a plant. They identified and altered an enzyme to create less lignin in the woody structure, thereby creating biofuels that are more easily extracted from woody plants. The hydrolysis of ATP is a way of creating energy for the cells in your body. ATP exists in your body. It is hydrolyzed to ADP, and in the mitochondria of your cells, ATP is remade. Let's look at a video that describes the formation of ATP and the energy it creates.
Oh, hello there. I'm at the gym. I don't know why you're here, but I'm going to do some push-ups, so you can join me on the floor if you want.
No, I'm not doing this just to show off or anything. I'm actually doing this for science, okay. Ugh. You see what happened there?
My arms moved, my shoulders moved, my back and stomach muscles moved, my heart pumped blood to all of those different places. It's pretty neat, huh?
Well, it turns out that how we make and use energy is a lot like sports or other kinds of exercising. It can be hard work and a little bit complicated, but if you do it right, it comes with some tremendous payoffs. But unlike hitting a ball with a stick, it's so marvelously complicated and awesome that we're still unraveling the mysteries of how it all works. And it all starts with a marvelous molecule that is one of your best friends, ATP.
Today I'm talking about the energy and the process our cells and other animal cells go through to provide themselves with power. Cellular respiration is how we derive energy from the food that we eat, specifically from glucose, since most of what we eat ends up as glucose. Here's the chemical formula for one molecule of glucose. In order to turn this glucose into energy, we're going to need to add some oxygen, 6 molecules of it to be exact. Through cellular respiration, we're going to turn that glucose and oxygen into 6 molecules of CO2, 6 molecules of water, and some energy that we can use for doing all of our push-ups. So that's all well and good, but here's the thing. We can't just use that energy to run a marathon or something. First, our bodies have to turn that energy into a really specific form of stored energy called ATP, or adenosine triphosphate. You've heard me talk about this before. People often refer to ATP as the currency of biological energy. Think of it as an American dollar. It's what you need to do business in the US. You can't just walk into a Best Buy with a handful of Chinese yen or Indian rupees and expect to be able to buy anything with them, even though they're technically our money. Same goes with energy. In order to be able to use it, our cells need energy to be transferred into adenosine triphosphate to be able to grow, move, create electrical impulses in our nerves and brains, everything. A while back, for instance, we talked about how cells use ATP to transport some kinds of materials in and out of its membranes. To jog your memory about that, you can watch that episode right here. Now, before we see how ATP is actually put together, let's look at how cells can cash in on the energy that's stashed in there. Well, adenosine triphosphate is made up of a nitrogenous base called adenine with a sugar called ribose and 3 phosphate groups attached to it. Now, one thing you need to know about these 3 phosphate groups is that they are super uncomfortable sitting together in a row like that, like 3 kids on a bus who hate each other all sharing the same seat. So because the phosphate groups are such terrible company for each other, ATP is able to do this nifty trick where it shoots one of the phosphate groups off the end of the seat, creating ADP, or adenosine diphosphate, because now there are just two kids sitting on the bus seat. In this reaction, when the third jerk kid is kicked off the seat, energy is released. And since there are a lot of water molecules just floating around nearby, an OH pairing, that's called a hydroxide, from one of the H2Os comes over and takes the place of that third phosphate group, and
metals are generally beneficial for human health in trace amounts. Trace amounts means not a lot. And we have, through our society, created the opportunity for our bodies to ingest more than trace amounts. There are good metals for the biological systems. Iron, for example, in heme, and it is also a part of ATP production. Zinc, it helps in many enzyme catalyzed reaction. Sodium and potassium, all important in generating nerve impulses. But there are some bad metals. These bad metals enter the food chain as one organism feeds upon another. And these metal ions can interfere with enzyme function by disrupting the disulfide groups and the hydrogen bonding. The metals can also disrupt van der Waals forces. Now, here are some examples of heavy metal pollution. By far, roadways and automobiles can be heavy metal producers. Zinc, copper, and lead are three of the most common heavy metals released from road travel. Industry also creates heavy metals as byproducts or as part of the processing chain, and these can get into the environment. But lead can come from gasoline, tire wear, oil and grease, wheel bearings. Zinc from tire wear, motor oil, brake emissions. Copper, all the things that help to make a car can have an effect. Cadmium, chromium, and nickel, as well as aluminum, all can have an effect on mammals. And once heavy metals are introduced into the environment, they normally cannot be biodegraded. They must be reacted with something else and removed from the environment. If not, they cause pollution of the air, the water, and the soil. Now, as you can see in this slide, there are a number of possible health issues that can be affected by heavy metals and heavy metal poisoning. Silver is a simple example. Silver takes the place between the hydrogen and sulfur bond because there is not that much electronegativity difference between silver and sulfur for a full ionic bond. And so the bond becomes covalent, and it disrupts the enzymes that are affected by these hydrogen bonds in the protein. I thought I'd give you a question to start your thinking at the end of the topic on biochemistry as to what careers might be available for chemists. We have spent probably 4 years studying inorganic inorganic chemistry. This is the first time in your 12th grade year that you've actually studied biochemistry. It might be advantageous for you to look and see what kinds of careers might be available to you.